everyone. My name is Carina Motta, and I'm a second-year pre-bio major here at UCSB. This summer, along with my mentor, Devin Orr of PI Hillary Young's Research Lab, we studied the effect of biodiversity loss and climate change on tick-borne disease. The first thing I want to talk to you guys about is how large land mammals, such as deer and elk, are the first to disappear when land use change occurs, such as urbanization or land is converted for agricultural use. As the abundance of these large land mammals decreases, it results in an overall increase in the vegetation in the environment. This in turn affects the remaining inhabitants of the ecosystem, such as the small vertebrates like squirrels, mice, and lizards. As this process is occurring, another process is simultaneously happening: climate change. As levels of carbon dioxide, methane, and other greenhouse gases are increasing in the Earth's atmosphere, we are seeing an increase in overall world temperature. These seemingly unrelated topics converge to have one thing in common: the tick. The small vertebrates serve as hosts for the ticks, and ticks are heavily reliant on ambient temperatures due to the fact that they are cold-blooded invertebrates. Now, ticks are vectors for disease, meaning that they can carry and transmit diseases to humans, such as Lyme disease. More humans contract Lyme disease per year than humans that contract malaria, HIV, and West Nile virus combined. Thus, this overall decrease in large land mammal population, combined with climate change, could potentially impact human health. My research this summer focused on the small vertebrates. Starting with the small land mammals, these small land mammals consume seeds as part of their diet, which in turn affects the reproductive ability of the vegetation. They also directly impact the amount of vegetation by consuming the plants themselves. Now, this can affect a tick due to the fact that ticks prefer a certain amount and type of vegetation. If the small land mammals are consuming this type of vegetation, whether in seed or plant form, they are decreasing the amount of viable tick habitat. That brings me to my first goal this summer, which is to study the impact small land mammals are having on vegetation in the ecosystem. Another thing I will be looking at is host-tick interactions and how small vertebrates, including lizards, serve as hosts for ticks. Since ticks are small and often hard to catch, it is easier to capture their hosts instead and use their host as a gauge for tick population, as well as zoonotic disease prevalence, which is my second goal. Zoonotic disease is a disease that can be transmitted from an animal to a human. So my research this summer was conducted at Tejon Ranch Exclosure experiment. We are located here at the red dot, UCSB, and Tejon Ranch is shaded in orange right here. It's 270,000 acres of preserved wilderness / cattle ranching. And so what's really interesting about Tejon Ranch is it actually is at the center of the convergence of 4 different ecosystems: Great Central Valley, Mojave Desert, Sierra Nevada, and South Coast. What this results in is a 33 to 178 centimeter per year rainfall range, as well as 120 to 2,020 meter altitude gradient throughout the whole range. What this allows us to do is set up experimental plots at different microclimates across the ranch. Our first is North Plot, which is at 2,800 feet in elevation. It's fairly flat and mainly composed of oak woodland. Our second is designated as Central. It's between 51 and 5,400 feet in elevation. It is mainly chaparral type ecosystem with scattered oaks and rubber rabbit brush. Our third is South, which is 53 to 5,600 feet in elevation. It is mainly dense black oak forest, and despite being close in elevation to the Central, is radically different due to the fact that it faces north and is much shadier. So at each of these experimental locations, we have plots set up, which are 100 meter by 100 meter, about the size of a football field. At each of these 9 plots, we have cafeteria trials set up. So there are 3 identical cafeteria locations at each plot, and at each of these locations is 3 petri dishes set up with different mixes of food options. So one of the first mixes is native mix, which is a mix between canyon oak acorns and pine seeds. The second is wild bird seed mix, which is just a standard mix purchased from Petco with millet and other small seeds. And the third is another non-native mix of corn and sunflower seeds. And so these were set out and checked periodically and reset a total of 4 times. In addition to conducting cafeteria trials, we also conducted small mammal trapping by setting up a 35 by 35 meter grid and setting up a total of 49 traps, 5 meters apart. The traps that we used were Sherman traps, which are essentially these small metal boxes which are triggered when an animal enters them to retrieve the food. In addition to that, we also hand-caught western fence lizards. So from the cafeteria trial results, we were able to determine herbivory rates at each of the 3 different locations. So from the north, we saw a fairly low herbivory rate, as well as a high preference for canyon oak acorns. In central, we saw a slightly higher herbivory rate and almost no preference when it came to food type. In the south, we saw a very high herbivory rate, as well as a slight preference to pine seeds. Taken as a whole, you can see a positive correlation between herbivory rates and altitude. And this is due to the fact that California has been in a drought for the past couple years, and at higher altitudes, there's more water available and less water stress on populations, allowing them to be more productive. And as a result, they are more competitive for food. For small mammal trapping, we had a fairly low success rate due to the fact, as well as the drought and a limited number of trials that we were able to conduct. So we only were able to trap small mammals at the south plot, which confirms our cafeteria trial data, which stated that herbivory rates were highest at the south due to high mammal abundance. The 2 small mammals that we captured were deer mice, one being shown here and being weighed after capture. In addition, we also sampled western fence lizards from the central plot, and so we captured a total of 6 and found that 3 of them had ticks on them. One had 2, another one had a total of 7, and a third had 3 ticks. And so now these ticks will be used, they will be used to, we will run PCR on them, which is a type of DNA amplification, so that we can determine whether Lyme disease is present, and, as well as other pathogens. As for future work, throughout the summer, we have also been constructing these small land mammal plots, exclosure plots, which are 5 meters by 5 meters and set up within the 100 meter by 100 meter ones. And so what we hope to find from these is very long term. We hope that by excluding these small land mammals, we will be able to see the impact that their absence has on the vegetation. In addition, we will be expanding our target audience for small mammal trapping to larger small animals. By using the Sherman traps, we only targeted small land mammals that were active at night, and these ones would be active during the day too, as well as larger. I'd like to thank EUREKA, as well as the EE&B department for funding, as well as CSAP and CNSI, without which EUREKA would not be possible. I'd also like to thank Professor Hillary Young and all her lab mates, as well as my mentor, Devin Orr, and my fellow ecologist, Anna Bell. Thank you. Questions? Yeah, Andy?
Did you harm any animals during this experiment?
No, they were all released after capture, after being tagged. Stephanie?
So can you go back to your data where you showed this north, central, and south field area? Okay, so in the south, it also coincides with the capture of 2 small rodents, but you also didn't find rodents in the central or north, is that correct?
Correct.
So could it be the reason why you're seeing such a high percentage of food consumption, could it also be like correlated to a higher population of rodents?
Yes,
And not
exactly.
necessarily-
So there's a higher population of rodents due to the fact that the ecosystem there is more supportive of them because there's more water available. So then since there are more rodents and a higher reproductive rate, there's more competition for food.
So, okay, if that's, so what was the logic with comparing like native versus non-native?
Basically to give us more data set points just so that we could confirm our findings rather than just using just acorns for ponderosa seeds.
But essentially you would expect that the same trend would have been seen in the acorn or ponderosa seeds regardless because it's just substrate or food.
Exactly.
Okay.
We're actually going to be exploring the reason why there was preference in some areas. For example, the canyon oak acorns were preferred in the north. It's possible that it's either due to high nutrient content or due to the fact that this is a recognizable food source and they prefer that.
Okay, well, so you were talking, so you set up the whole talk with understanding that these are carrier for diseases that ticks carry.
Right.
So I know this is pretty preliminary, but can you talk to the fact of, are you seeing more ticks in the south versus the north as a consequence of just more population, or if it's actually each rodent density has a more higher?
We'd have to run more trials to have a concrete conclusion, but yes, for now we are seeing more small land mammal populations in the north, and actually we saw the highest lizard population in the central, and that's why we caught the lizards in the central.
What's the connection with the lizard to the rodents?
So the lizards and the rodents are both hosts.
So did you also capture lizards
Yeah.
in the same, what fancy method that you use?
No, not using Sherman trapping method, hand caught, which I showed here.
And so you can caught them in the same grid zones, or
Yeah.
were these completely independent?
Same grid zones.
Okay.
Amanda?
Did you find ticks on the deer lines?
No. Emma?
How did you count the ticks? Like, what process did you go to prevent, contamination?
Oh, well, we picked them off one by one and put them in an O-ring container.
So you went through and searched individually and picked them off.
Yeah.
All right.
Jason?
How many ticks did you get?
0, hopefully. Yes?
I was wondering if one of the goals of the project is to compare whether the fact of having more hosts, what the effect of having more hosts is compared to the effect of the hosts eat the vegetation. Did you get any information about how those two influence the actual tick population?
Not yet. That's part of our future plans as well as for doing the small land mammal exclosure plots. But we do know that ticks are density dependent, and therefore the more small land mammals or small vertebrates, the more ticks there will be because there are more hosts.
Thank you for